In 2012, a new member of the powdery mildew genus Erysiphe was documented in various hazel tree species in the genus Corylacearum from East Asia and North America, where it is considered to be endemic, designated as Erysiphe corylacearum. Although visually similar to the other powdery mildews like the fairly benign Phylactinia guttata, known for colonizing the stems and leaves of hazelnut trees, this new species also colonized the young shoots and nut clusters, making it much more destructive to both cultivated and forested trees. In the spring of 2013, Erysiphe corylacearum was documented infecting hazelnut orchards in the Black Sea region of Turkey, with severe damage sweeping through from east to west starting in Trabzon and expanding into two of the largest hazelnut-producing provinces, Ordu and Giresun. Colonies of the species develop on the underside of leaves and cause visible mottled pale or yellow discoloration of the upper surface. The leaves tarnish as lesions turn brown in color, and sexual fruiting bodies called chasmothecia develop. Infected leaves and nut clusters dry and curl, falling early with sensitive cultivars afflicted with significant crop losses. This serious threat to worldwide hazelnut production was identified, and the Republic of Turkey commissioned research efforts into containing it. In the July growing season of 2016, all 52 groves sampled in these three provinces had the new hazelnut powdery mildew, with leaf infestation severity hovering around 87% for most cultivars, and nut cluster infestation severity at 45% or so. 6 commercial cultivars were sampled: Minshan, Fosha, Sivri, Chakildak, Palaz, and one of the most widely cultivated and highest quality cultivars, Tombul. Tombul, and to half the extent Palaz, had the most incidences, but Palaz had the least incidence of nut cluster disease severity. Other cultivars sampled that had lower instances of leaf and nut cluster disease severity included Uzun Musa, Khan, Yuvarlak Badem, and Inchakara in one study. Hazelnuts from Turkey account for approximately 70% of the world market and are the country's single most valuable agricultural export, with annual sales above $1 billion in 2011, according to a 2017 report by the Food and Agriculture Organization of the United Nations. World production in 2017 was 1 million metric tons of raw hazelnuts, with Italy commanding approximately 15% of the market, ahead of small single-digit percentages in Azerbaijan, the USA, and China. Close to 61% of Turkey's rural population depend on hazelnut harvests for income. Additionally, most of the country does as well. Current populations of the common, or sometimes called European hazelnut trees, Corylus avellana, originate in Asia, after recolonization following the last glacial maximum approximately 15,000 years ago. Mesolithic human and animal populations between 10 to 6,000 years ago fed on hazelnuts extensively, which is thought to have led to incidental dispersal, whereas intentional cultivation likely occurs several millennia afterwards, with multiple sites of independent cultivation and domestication across the Eurasian continent, most prominently in Western Europe and Central Asia, though genetic admixtures have been documented occurring throughout this time. While annual crops tend to rapidly become domesticated due to their shorter lifespans and life cycle cycling, perennial crops like orchard trees are not always subject to domestication despite being cultivated, and in a recent study, sampled hazelnuts were not found to have a particular domestication bottleneck. Although often propagated through clonal cuttings, genetic diversity assessments of Turkish hazelnuts found that, for example, the plants referenced as tombul in the field that are known to be the most economically important cultivar were genetically quite disparate. This may be because cultivar names can be based on traits that are not correlated with neutral genetic variation such as kernel size, shape, or taste. Morphology has been used to assign Turkish cultivars to 3 primary groups, primarily based on nut shape in other studies, but these do not correspond to the genetic clusters recovered. Since resistance genes are integral for resistance against this hazelnut powdery mildew, disparity must be resolved in order to understand the strengths and weaknesses of the global hazelnut genome. For similar ecological reasons, crop-to-wild crossings can affect wild populations and cause extinction in such populations since hazelnuts are self-incompatible, which facilitates hybridization. Sampled Erysiphe corylacearum populations show great similarity at the genomic level when DNA barcoded, and analyses have shown the current global outbreak is due to a single recent transmission event. In fact, in the last 2 decades, powdery mildew species have been documented to move from east to west and established throughout the Eurasian continent, starting in Ukraine, Turkey, Romania, and moving westward through both natural and maintained landscapes. Though there are generally disparate genetic clusters of European hazelnut throughout its range, genetic similarities born from clonal propagation may increase the likelihood of finding resistance material for agrarian projects, though simultaneously, this genetic similarity also means that at the orchard and inter-orchard level, hazel populations are likely to share the detriments of genetic susceptibility as well. Since 2013, the virulent strain of Erysiphe corylacearum has been documented in Azerbaijan, Iran, Georgia, Switzerland, and as of September 2020, the Austrian provinces of Burgenland, Carinthia, Lower Austria, Styria, and Vienna. Efforts to propagate resistant cultivars in the face of this destructive plant parasite are active and ongoing. Typical treatment measures for the powdery mildews in agriculture, besides the use of resistant populations, include fungicidal chemical agents, biocontrol agents, as well as changes in harvest or production technique. Ampelomyces quisqualis is well known as a natural hyperparasite of more than 64 powdery mildews, and a potent biocontrol agent for many species. Research in the western Black Sea and Marmara regions of Turkey was conducted in July and August of 2016 and 2017 to measure the presence of Ampelomyces populations generally in hazelnut orchards afflicted with Erysiphe corylacearum. The dank, rainy temperate climate of those regions were favorable for Ampelomyces, which requires free water and warm temperatures of 20 to 30 degrees Celsius for successful infection. Incidences in orchards were high and implied that both hazelnut powdery mildews are affected by the hyperparasite, which may become a formidable agent of biocontrol. The Ampelomyces quisqualis strain CPA-9 has been shown to be effective against certain powdery mildews, especially with high conidia, or asexual spore counts. For practical use to be viable, however, a high number of infective material must be produced and dispersed at the correct frequency and rate. Many powdery mildews reproduce asexually, but are still able to adapt to new environments and host resistances through a special kind of mutation called repeat induced point mutation, which can facilitate the production of more genes, and especially those directly related to virulence, without sexual combination. Regardless of this isolate of Erysiphe corylacearum is often described producing chasmothecium, sexual fruiting bodies, and this may influence adaptation in the future or explain its recent outbreak. More information on the origin of powdery mildews and how they are thought to have developed from decomposer fungi that gained virulence traits can be found in my video, Why is Powdery Mildew?